things that are important, things that are interesting, and things that are surprising. We encourage our studio audience to ask any questions that you have. Please come up to the microphone to ask questions. And if you're listening in online, you can send questions to #ASM2014. Our guests today are Jessica Rivera Perez from the University of Puerto Rico in Rio Piedras and Raul Cano from Cal Poly San Luis Obispo. Oops, sorry about that.
Happy and well.
And they have a collaboration between the two campuses to study fossilized species and how fossilized species can help us understand pre-Columbian cultures. Who would have guessed? Jessica, could you tell us a little bit about how you even began to think about doing this study?
Well, the idea actually came from Raul. He got in contact with the archaeologists Yvonne Narganes and Luis Chanlate from the Center of Investigations in Archaeology in our campus in the University of Puerto Rico. He found out that these archaeologists had access to these coprolites, these fossilized species, and he basically stuck his neck out and the collaboration began. And then he began incorporating other scientists, bioinformatics, Dr. Steve Massey, the microbiologist, for example, Dr. Gary Toranzos, which is my mentor. And so we began trying to see if we could characterize the fecal microbiota of two cultures, two pre-Columbian cultures that had inhabited the island of Vieques, which is in the eastern side of the mainland in Puerto Rico in the Caribbean. And so these cultures were a bit of a mystery up to a certain point. One of the cultures was well characterized, the Saladoid culture, but the second one, it was still unknown if they had the same origin of the Saladoid culture. And so since they weren't sure where they came from, they gave it the name Huecoid in honor of the place where the excavation site was located in the 70s. So what we did was basically characterize the fecal microbiota and see if we could determine major differences between these two cultures.
So Raul, what got you into it? What made you think about fossilized species to begin with?
Well, besides being a fossil myself, I've been working on fossils since 1992, 93, and for a while, funding became kind of an issue, so fossils are not fundable. So unfortunately, we had to move on to bioremediation because that's what the action was at the time. But I retired in 2005, and I still have a passion for ancient DNA. So excuse me, this opportunity presented itself. Dr. Narganes was kind enough to let me share some of her jewels, and so that was a perfect, for me, that was a perfect retirement present.
Jewels, present, feces, there's just something incongruent about this.
I am not particular.
So you have these two cultures. Were they physically separate enough so that you could distinguish which fossilized species came from which particular one of these groups?
It is my understanding from looking at, discussing it with the archaeologists and also looking at the evidence that they co-inhabited the island of Vieques. Actually, their excavations were pretty close to each other, within walking distance. But they were still remain distinct and identifiable based primarily on archaeological remains.
They had different outhouses.
Yes. Well, I presume so, which is close to the garbage dump.
Dr. Narganes, she actually at one point commented that they were actually living a maximum of 20 meters apart from each other, but they were very separate. one culture had one part of the region to themselves, and then the other culture would have the other area of the region.
That's very interesting, just from a social perspective. But now, why don't you tell us what you can learn from looking at these feces? First of all, how do you analyze fossilized feces?
With patience. Well, first, what we did was we actually tried to extract the DNA various times. And at first it was kind of uphill, but we actually got enough DNA for sequencing. And we did the bioinformatic analysis, and what we found was pretty amazing. I mean, the quantity of DNA that have been preserved in these species, I don't think we were even expecting such good quality DNA, and since these coprolites are not supposed to be well preserved in the tropics, then it was just an added surprise.
But to add to this, our laboratory has had upwards of 20 years of experience working with ancient DNA. So we know what the caveats are. And we have tried over the years to improve on the quality and the handling of the DNA to avoid obvious problems that you may encounter with contamination, etc. And knowing that the DNA really is sparse at best, one has to do specific methods to be able to separate the DNA and ascertain that the DNA is actually from the origin, which we hope it's from. So, yeah, we've had DNA difficulties extracting because they're not much, but the quality is excellent, and it has a lot to do with technique and experience.
So presumably you have DNA from microbes that are in the feces, you have some DNA from the host, and potentially some DNA from other things that the host consumed in their diet.
That's exactly what we're observing in the data.
And tell us, what did you learn the most from?
The most?
Well, so for example, one of the key things that you would like to know is differences in the diet of these two populations. Is that something you simply infer from looking at DNA signatures of plant products, or do the microbes also tell you something about that?
We kind of put both things together because previous studies have demonstrated that the fecal microbiota and the gastrointestinal microbiota are influenced by the diet of the host. But also, if you're detecting, for example, in our case, we detected maize in one of the cultures. Well, I mean, it's obvious how that maize got into that feces. So you put both data sets together and you start trying to develop a story.
But the approach has to be systematic. Okay, so first of all, the first question is, are there microbes in here? Or is there any DNA in there? And once that question is answered, then you go to the next step. It says, you set up the null hypothesis. There's no difference between the DNA from all of these cultures. And then once you either prove or disprove that null hypothesis, which in our case was, no, they are not the same, they are different, then you start asking for deeper and deeper questions and separate it microbiologically, bacteria, fungi, viruses, and functionally, and carbohydrate digesting enzymes, lipid processing enzymes, things like that.